right, it's a pleasure to be here, and thank you for hanging in on a beautiful day. So, as the title tells you, Perfectus Vaccines practices in the areas of enhanced DNA vaccination. We have a recombinant vesicular stomatitis virus-based vector platform, and we use those in conjunction in prime boost immunization. And we've recently been finding a lot of data which indicates that it's a very effective combination for steering immune responses. And by that I mean inducing responses of a high, not only high magnitude, but also that are qualitatively quite attractive. So why think about quality in addition to quantity of your immune response? These are some of the early papers which came out starting about 2006, which indicated that in a series of chronic infections, really the quality of the immune response was as important as the quantity in being able to control. So the first paper that came out, which was really pointed in this direction, looks at non-progressors that are HIV infected and shows that their CD8 cells bear a particular phenotype and that they're multifunctional. They're able to secrete multiple cytokines. And that's been built on now. There's a very large body of literature which is being built up. So in HIV, CMV, here's Leishmania, and a number of other chronic viral infections, including HCV, HBV, these data are accumulating. All right. We didn't get, okay, I'll go backwards. All right, so here is a non-human primate study, 6 rhesus macaques per group. We are looking at an HIV DNA vaccine encoding envelope gag, pol, neftat, vif. And we are looking at how we can induce an immune response of substantial magnitude and an immune response which has particular qualitative aspects which we're seeking. We want to match what we see in individuals that are long-term non-progressors. So this is a study in which we use the DNA vaccine alone. It was delivered using electroporation. Here is the DNA vaccine in co-formulation with a plasmid encoding interleukin-12 as a molecular adjuvant. And here is the DNA vaccine when it is co-formulated with the interleukin-12 plasmid and delivered by electroporation. So the increase in the magnitude of the immune response is obvious, which is desirable. Statistically significant from either of the two components alone. But in addition to that, you're able to look at the quality of the immune response. So you do that by doing intracellular cytokine staining, and 11 parameter flow cytometry. What you do is you would gate on live cells, gate on the lymphocyte population. You would stain for CD3 so that you're only looking at T cells, stain those T cells in addition for CD4 and CD8, and then you can fix, permeabilize. You've re-stimulated them with a peptide pool. The responding cells will begin to secrete cytokines, which you block internally. You then stain them internally for interferon gamma, IL-2, MIP-1 beta, and interferons as a method of looking at their ability to secrete multiple cytokines. All right, so here is the readout. We're looking at cells which are secreting one cytokine, interferon gamma, IL-2, so on and so forth, any combination of 2, any combination of 3, or 4 cytokines, and they're color-coded. Red indicates cells which are secreting a single cytokine. The green tells you the cells are secreting 2, blue 3, and so on and so forth. Here are your pie charts. Inside are the absolute magnitude of the responses. In this case, we're looking at CD8 cells. They're in the same group of monkeys we saw the increased response levels. We're looking at the gag-specific response in these animals. What you see is that the combination of electroporation plus IL-12 not only provides you with the response to the DNA vaccine of the greatest magnitude, the greatest breadth, but it also imparts the greatest increase in the quality of the response in that you have cells of the phenotype which are have multiple potential to secrete cytokines, and those are exactly the types of responses that you want to see in an individual where you're immunizing against HIV or HCV or any other chronic infection. Okay. Now, how are we doing at translating these responses we see in non-human primates into clinic? In the first step, the interleukin-12 plasmid in combination with electroporation was the subject of a recent HVTN trial, the 080 trial. It looked at 3 doses of a Penvax-B, a DNA vaccine similar to the one that I just described to you, which comes out of the laboratory of Dr. David Weiner. It was given to 40 healthy HIV-negative volunteers. 10 received the DNA vaccine with electroporation. 30 received the vaccine in combination with the GeneVax IL-12 plasmid. And the responses were measured in an ICS, an intracellular cytokine staining assay, similar to the one that I just described to you, with a readout of either interferon gamma or IL-2 responses upon peptide restimulation. The interim data, which have been reported on in the last month or so, 38 out of 40 post-dose 2, 31 of 40 post-dose 3. After 2 vaccinations, a 30% response rate to the DNA vaccine given with electroporation alone, a 71% response rate for those receiving also the GeneVax IL-12 that reaches statistical significance. After 3 vaccinations, 6 of 9 with DNA and electroporation alone responded, but 91% also receiving the interleukin-12 responded, and that gets you up to a response rate of 91%. When you look at the CD8 component of that response, you find that the CD8 CMI response is double anything seen in any other trial conducted by the HVTN. So we are managing to translate some of these non-human primate data into clinical validation. Okay. The second part of the vaccine platform is vesicular stomatitis virus. Interesting virus, single-stranded, negative sense RNA virus, simple genome. The position of the genes in the genome determine the level of protein expression. There's a transcriptional gradient. So as the transcriptional machinery reads down, comes to an intergenic site, it has about a 90% chance of falling off. That means that the N protein is made in greater abundance than P, M, G, and L, and so on and so forth. You can genetically manipulate this virus. If you put your transgene in the first position, you'll make 10 times as much of the transgene as any viral protein. So it's a system which is nice because it actually increases the expression and response to the transgene, but not at the expense of making a strong antivirus response. So it's an RNA genome. It's small with few proteins, but a very large capacity for multiple gene inserts. There's a transcriptional gradient, as I've explained. It's an RNA virus, so there's no risk of integration. Human infection is largely asymptomatic or mild. That's with a wild type. We have a clinical vector which has been attenuated through multiple genomic modifications, which is currently the IND is submitted, and we should be free to proceed into the clinic on the 21st. Let's see, immunogenicity, it's replication-competent virus. It targets antigen-presenting cells and triggers multiple innate immune pathways through various mechanisms, including the creation of a double-stranded RNA during its replicative cycle. It's a proven delivery system. It propagates very efficiently in continuous cell lines, and we are currently at a practical commercial-scale manufacturing. 10-liter bioreactor will yield 50,000 to 500,000 doses of finished vaccine. Okay. In addition, there's little pre-existing immunity in the human population, and many serotypes are available, so you can switch out the G protein and get around pre-existing immunity and neutralization against your vector. All right. So here's actually an ongoing study It's actually looking at an HCV vaccine which encodes the NS3, 4A, 4B, 5A, 5B components, the non-structural proteins of HCV. It's a simple study. It's asking the question whether heterologous prime boost really delivers with respect to both magnitude and quality of immune responses. So you're looking at 6 macaques immunized with HCV plasmid DNA with IL-12 delivered with electroporation, as I've showed you previously. They get a 3 vaccine priming regimen, and then they're boosted either with another dose of the DNA vaccine with IL-12 and EP, or with a VSV vectored vaccine construct which has the same non-structural proteins in it. All right, so here are the immune responses as far as magnitude is concerned. The DNA prime VSV boost versus the DNA prime DNA boost. Here are the components to the vaccine, the response magnitudes, and you can see that the prime boost combination, as we saw previously, is substantially more able to induce very, very robust immune responses. In addition to that, when one starts to look at the qualitative nature of those responses, you can see that the VSV boosting actually further potentiates the type of response that you're looking for. All right, so here are the same sorts of plots. DNA, which are the red bars, DNA VSV, which are the dark bars. The magnitude of the response is shown here, and the pie charts read out in the same way. 1, 2, 3, and 4 cytokine secreting cells. 40% of the response in the CD8 population is multifunctional when you use DNA, but if you use a VSV booster, you can increase that to about 70%. This is the response profile which is seen in long-term non-progressors. This is what I want my vaccine to do. Okay, you can dig down a little bit deeper by looking at central memory and effector memory populations. Here again, the DNA versus the DNA VSV response magnitudes go up, and the percentage of cells in both the central memory, long-lived memory cells, as well as the effector memory cells, the cells that are doing the job right now, the polyfunctional nature is dramatically increased from 20 to 64% and from 48 to 73%. If you go on and look at the CD4 population, you get the same answer. The DNA VSV boost provides a response which is of, in this case, approximately the same magnitude. However, the percentage of the cells with this multifunctional capacity is very substantially increased. Again, if you drill down into the central memory CD4 cells and the effector memory CD4 cells, those effects hold. So, in summary, you start working with DNA vaccines, you're able to adjuvant the responses to DNA vaccines with things like cytokines. IL-12 is the one we have the most experience with, and drive not only a response of greater magnitude, but also a response of a better quality. So we saw the enhanced both CMI and antibody responses in non-human primates, and we've seen improvement in the polyfunctional CD4 and CD8 responses in non-human primates. Our clinical data now from the HVTN 080 trial shows that we are able to improve human immune response rates and magnitudes by using interleukin-12 as a vaccine, as a molecular adjuvant, and the human polyfunctional CD4 and CD8 responses are being measured currently, and I'll predict that we'll see an improvement here. pDNA prime with IL-12 with electroporation followed by VSV boosting very strongly enhances rhesus CMI and antibody responses to levels that are the highest we've ever witnessed. And it also dramatically improves the polyfunctional nature of both CD4 and CD8 responses, their longevity, how long that memory will last. So what are our next steps? The ACTG 5281 trial in HIV-infected individuals is enrolling right now. That's a trial of a multi-antigen plasmid DNA vaccine from Perfectus with a dose titration of plasmid IL-12 when delivered by electroporation using the iCore Medical Systems electroporation device. So that's starting up. We have 4 patients lined up, and we'll start vaccinating next week. The HVTN 090 trial is a first-in-man trial of a VSV vectored vaccine. It's attenuated by shuffling the N protein to the 4th position and truncating the cytoplasmic tail to a single amino acid. The dose titration will be 10 to the 4, 5, 6, 7, and 8 for safety reads. The IND was submitted on the 23rd of March, and again on the 21st of this month, that'll be an active IND. FDA sent us their questions. They're quite benign at this point. The HVTN 087 trial is a trial of multi-antigen plasmid DNA with plasmid IL-12 delivered by EP for a prime, followed by that VSV boost. That tox study is complete. It's clean. The protocol is largely done and in review, and a IND start time is targeted for mid-4th quarter of this year. So we'll see how the non-human primate data are going to translate across the board. So the acknowledgements. This work originally initiated at Wyeth and has subsequently been transferred out to Perfectus Biosciences. Thanks to many people at Wyeth, those with an asterisk after their name actually moved over to Perfectus with me to continue these studies. We have the DNA vaccines have been modified in the Pavlakis laboratory to RNA optimize them, so thanks to the National Cancer Institute. The Division of AIDS, which has been highly supportive of all these programs. iCore Medical Systems, whom we currently work with their electroporation device. Much of the non-human primate, all of the non-human primate work was conducted at the New Iberia Research Center. Acknowledge John Rose at Yale University as the man who identified the capacity of VSV to be a vector. The ACTG for conducting trials for us. The HIV Vaccine Trials Network for conducting trials for us. David Weiner was a collaborator early on with respect to the cytokines, and the original studies that we conducted on electroporation used the Inovio device. So with that, I'll close and open for questions.
So very exciting stuff. A couple of questions. It's a question because I was looking at John Dung's earlier data, and with the use of cytokines to act as adjuvants. As you introduce IL-12, and I can't recall if this was a single exposure of your plasmid for the prime, or are you continuing to boost additionally? So my question is around the cytokine itself that you're inducing. Are you being asked to look at antibodies to IL-12 after it's being produced?
Are we being asked to look at antibodies against the IL-12?
Yes, so if you look in the biological world where you're trying to develop therapeutics, yeah. Almost all of those compounds do have anti-drug antibodies. So as you introduce this molecule into the system, the question is whether or not you might have an anti-cytokine antibody.
It's fully natural IL-12. We've taken it into the clinic multiple times without electroporation. We've measured anti-IL-12 antibodies in all those subjects. We've never seen it before. We'll also measure anti-IL-12 antibodies in this trial, okay. But I don't expect to see anything.
So there was a lot of interesting data on magnitude and polyfunctionality. Did you also have an opportunity to look into breadth and depth of the immune response? And especially with regard to depth, does this technology allow to have a shift in the immunogenicity of the individual CD8 epitopes? So what I mean with this is that CD8 epitopes that are not naturally immunogenic and subdominant, does this technology enable to make them more immunogenic?
Okay, so I think your question basically boils down to, do we manage to see a broadening of the immune response as well as just a an increase in magnitude. And yes, if you do the crosshatch peptide study and you look for how many epitopes are being responded against, you find broadening of the responses pretty much in the same way that you saw increases in what I labeled the quality of the immune response. So adding IL-12 increases the magnitude, it also increases the breadth of the CD8 response. Electroporation helps, the VSV boosting helps.
But what about that many of these epitopes are pretty much conserved but are not really immunogenic, which is a problem with, I would say, HIV. So there's this technology, so some of these conserved epitopes are not naturally immunogenic. Does this technology enable you to make some of these epitopes, which we know are not naturally immunogenic, to make them more immunogenic actually?
I mean, you can attack that from a variety of directions. And in this case, by definition, since we're broadening out the number of epitopes being recognized, we are increasing the breadth, and we are extending the responses to subdominant epitopes. I think your question is, would I like to go backwards and design an immunogen which contained, had the dominant CTL epitopes removed in order to drive the response towards subdominant epitopes. I think that's an interesting path to go down, and it could be quite productive in vaccine design in the future.